I think if you've done the gene-for-gene hypothesis, if you studied it during your courses, you would have heard of Flor. It was Flor that actually came up with this hypothesis back in the 1950s, I think. He studied flax rust. So he studied not just the genetics of resistance in the host, but he also studied the genetics of pathogenicity in the pathogen in relation to the genetics of the host resistance. Very clever work. No one had ever done that before. People had studied the genetics of resistance in the host. People had studied the genetics of pathogenicity in a pathogen, but no one had done concurrent studies of genetics of both. And that's what Flor did. So the first thing he did was that he found two host genotypes, two flax genotypes, and when he inoculated with one pathogen isolate, a pathotype, he found that one was resistant and the other one was susceptible. And he wanted to know what the genetic basis of this resistance was. The way he did that was he intercrossed the two to produce a segregating population. So he intercrossed host number 1 with host number 2. He got an F1, he selfed the F1 and went to the F2, and then when he got the F2 progeny, tested them, did a rust test with pathotype number 1. Very important. He used this pathotype that was low. I gave a low infection type on host 1 and a high infection type on host 2. And what he found was, roughly speaking, that for every susceptible plant in the F2, there were 3 resistant plants. A segregation ratio roughly of 3 is to 1. That very famous 3 is to 1 segregation that Grigor Mendel first reported for flower colour in P. And Neil was talking about this a bit yesterday. I think, Gordon, you were mentioning it as well. These segregation ratios that tell us how many genes are segregating. So the conclusion was, in this case, that host 1 carries a single dominant resistance gene against pathotype 1. That was the conclusion. Very basic, simple genetic study of the inheritance of resistance in host 1 to pathotype 1. That was the first part of the work that he did. What he then did was he found a second rust isolate. So we still have pathotype number 1, which we now are talking about the pathogen, right? So it is avirulent on host 1. And a second pathotype that was virulent on host 1. So here we have 2 different pathotypes that differ in their pathogenicity on that host genotype 1 that he'd previously shown carried a single dominant gene for resistance. And he wanted to know the genetic basis of that avirulence. So what he did was he intercrossed the 2 rust isolates and produced an F1 and then selfed the F1 to get F2 progeny of the rust pathogen, so multiple isolates of the rust pathogen, F2 isolates, and then he used those F2 isolates to inoculate host 1, and from that he found that for every isolate that was virulent, every F2 isolate that was virulent on host 1, on average there were 3 that were avirulent on host 1. All right? So, for example, he had 4 isolates, 4 F2 isolates. The first 3 were avirulent on host 1. The last one was virulent. So that's a segregation ratio of 3 avirulent to 1 virulent. And the conclusion, of course, was that pathotype 1 carried a single dominant gene for avirulence on host 1. All right? So here we have a system where host 1 carries a single dominant gene for resistance, the pathotype 1. Pathotype 1 carries a single dominant avirulence gene on host 1. Now the really amazing thing about this is when he did a number of these studies, he found that there was specificity, right? So the avirulence gene in pathotype 1, he found corresponded specifically to the resistance gene in host 1. It didn't match resistance genes in other genotypes that were different to the one that occurred in host 1. So there was a gene for gene relationship. A gene for avirulence in the pathogen matching a gene for resistance in the host. And that correlation was very specific. So he came up with a thing called the Quadratic Check. Okay, this is like a Punnett square, I suppose you'd say, but it shows possible host genotypes across the top for a single gene and possible pathogen genotypes for that single gene with respect to avirulence down here. I've not shown, it's simplified because I have not shown the heterozygotes here, okay? These are only the homozygous genotypes. We could have, for example, big R, little r, or big AVR, little AVR. I haven't put them in for simplicity, all right? And you'll see from this that it's only when we get that matching resistance gene and avirulence gene that we end up with incompatibility or low infection type. In all the other possibilities, here the avirulence gene is present, but the resistance gene is not. We have susceptibility. Down here, where the avirulence gene is lacking, but the resistance gene is present, we have the high infection type or susceptibility. Down here, both the R gene and the AVR gene are lacking, and we end up with compatibility or susceptibility. All right? Now, if you want to think of that in, what is the molecular basis of this in terms of the actual physiology and what's going on at the molecular level, the work that's been done on cloning R genes and AVR genes supports this model, okay? So what we believe is that there's an interaction going on like a lock and a key, like an antigen-antibody interaction. So the R gene in the host is producing a protein. So you can think of this maybe as being a hypothetical protein being produced by this R1 resistance gene. The AVR gene in the pathogen is also producing a protein, all right? As depicted here by this green circle. What we believe happens is that those two proteins, or the host resistance gene protein, recognises the pathogen protein, right? Like a lock and a key. Very specific interaction between two gene products, one from the host, one from the pathogen. And it's that recognition event that triggers or switches on a whole stack of defence-related genes that then stop the pathogen growth and give us that incompatible interaction. All right, so the resistance gene is not the thing that's stopping the pathogen necessarily. It is actually a gene for recognition. It's recognising the presence of the pathogen because it's recognising that avirulence gene product, and it's then turning on all of these defence-related genes that then stop the pathogen growth, that give the hypersensitivity and all the other stuff, right? And you can see here that the specificity is dictated by the two gene products being able to identify each other like a lock and a key. And that recognition event then switching on the defence pathway in the plant. What happens down here, for instance? Well, we still have the resistance gene, all right? In this interaction here, the resistance gene is still there in the plant. It's still producing that protein. But the pathogen's protein for that avirulence gene has changed because the avirulence gene is, we've now got the alternate virulence allele here, and so the gene product has changed. So in this case, there's no recognition. So the plant cannot turn on its defence-related genes. There's no recognition of the presence of the pathogen. The plant can't switch on its defence-related genes, and the pathogen is free to infect. It got the green traffic light, if you like. It's infecting and off it goes, and we end up with compatibility. The other important thing to realise is sometimes you'll read in the literature people talk about virulence alleles in a pathogen. Okay, now there are such things as virulence alleles, but we also can get virulence from the complete absence of a locus, the corresponding locus in the pathogen. In other words, no gene product whatsoever. So the avirulence gene, the alternate virulence allele, is a null allele. It's not doing anything. It's not producing any gene product at all. So the pathogen is invisible to the host. There's no gene product there being produced by the pathogen. The plant's got no way of knowing it's there, so it's free to grow. Again, it gets the green traffic light. All right? So that virulence state in the pathogen can be a different protein, or it may be no protein at all being produced by the pathogen. It becomes invisible to the host and it can grow. Okay? So what that all means is that when we see incompatibility in a low infection type, if we take a rust isolate and we infect it onto a wheat plant and we see a low infection type, the gene for gene hypothesis implies the presence of at least one corresponding avirulence and resistance genes in the host and the pathogen respectively. So straight away, if we see incompatibility, we can deduce a host and a pathogen genotype. Right? Where we see compatibility or a high infection type, we don't know what the genotype of the host and the pathogen is. There are three possibilities, right? We don't know what the genotype is because it could be either this, it could be either this, or this, right? Three possibilities, and we don't know which one. All right, so when we see incompatibility, the gene for gene hypothesis allows us to deduce a genotype for the pathogen and the host. When we see compatibility, we can't. We don't know which one of those three possibilities is actually operational. And I think I've said that there in the yellow. If we know either and so the corollary of that, and this is where we can apply the gene for gene hypothesis in resistance, is that if we know either the pathogen genotype or the host genotype, the gene for gene hypothesis allows us to deduce a genotype for the host or the pathogen respectively whenever we see that incompatibility. Right? Now, that may sound a little confusing, but I'm now going to spend a fair bit of time explaining what I mean by what's in the yellow box there, all right? How are we going for time and lunch?
We've got an hour after lunch for discussions.
Yeah?
So I think we've got plenty of time for the subject. 10 minutes.
10 minutes. All right, so let's at least make a start. All right, so the gene for gene hypothesis basically gives us two, what you might think of laws like sort of like Mendel's laws, but not quite. Okay, two laws of genetics of host pathogen interactions. One is, this is law number one, a low infection type means avirulence and resistance. If you see incompatibility or a low infection type, then you know the pathogen must be avirulent, it must have a gene for avirulence matching a gene for resistance in the host, right? A high infection, and that's shown down here by this particular square here in our matrix, all right, A. A high infection type, on the other hand, could be AVR over susceptibility, which is this one, right? So here's the avirulence and here's the resistance, or it could be this one, or it could be this one. Compatibility could be any one of those three possibilities. So that's our first law of host pathogen genetics. A low infection type tells us that there must be at least one corresponding avirulence gene and R gene. Compatibility can mean one of three possibilities.